welcome to Biology, the scientific study of life. The preservation of genetic variation. What prevents natural selection from reducing genetic variation by killing all unfavorable genotypes? The tendency for directional and stabilizing selection to reduce variation is countered by mechanisms that preserve or restore it. Diploid. Because most eukaryotes are diploid, a considerable amount of genetic variation is hidden from selection in the form of recessive alleles. Recessive alleles that are less favorable than their dominant counterparts or even harmful in the current environment can persist because they are propagated in heterozygous individuals. This latent variation is exposed to natural selection only when both parents carry the same recessive allele and combine two copies in one zygote. This happens only rarely if the frequency of the recessive allele is very low. The recessive allele, the greater the degree of protection from natural selection. Heterozygote protection maintains a huge pool of alleles that might not be favored under present conditions, but some of which could bring new benefits when the environment changes. Balancing selection. Selection itself may preserve variation at some gene loci. Balancing selection occurs when natural selection maintains stable frequencies of two or more phenotypic forms in a population, a state called balanced polymorphism. This type of selection includes heterozygote advantage and frequency-dependent selection. Heterozygote advantage is individuals who are heterozygous at a particular gene locus have greater fitness, and then the homozygotes. Natural selection will tend to maintain two or more alleles at that locus. An example of this heterozygote advantage occurs at the locus in humans that calls for one of the peptide subunits in hemoglobin. The oxygen-carrying protein of red blood cells in homozygous individuals, a recessive allele at that locus, causes sickle cell disease. Notice this figure. However, heterozygotes are protected against the severest effects of malaria, although they are not resistant to malarial infection. This protection is an important advantage to tropical regions where malaria is a major killer. The environment in this area favor heterozygous over homozygous dominant individuals who are more susceptible to malaria, and also over homozygous recessive individuals who develop sickle cell disease. The frequency of the sickle cell allele in Africa is generally highest in areas where the malaria parasite is most common. Notice that. In some tribes, it accounts for 20% of the hemoglobin alleles in the gene pool, a very high frequency for such a harmful allele. But even at this high frequency, Q equal 0.2, only 4% of the population suffers from sickle cell disease. Q to the power 2 equal 0.044, which means 4 part of 100. There are far more heterozygotes who are resistant to malaria. 2BQ equal 2 by 0.8 by 0.2 equals 0.32. Although the fitness advantage to the heterozygotes is much smaller than the disadvantage to the homozygote, there are so many more heterozygotes than homozygotes that the aggregate benefit to the allele in the population balances its aggregate harm. Notice mapping malaria and the sickle cell disease. Notice also the sickle cell allele is most common in Africa. but it is not the only balanced polymorphism that offers protection against malaria. Different balanced polymorphism are found in populations in other parts of the world, such as around Mediterranean Sea and Southeast Asia, where malaria is widespread. Also notice that. Frequency dependent selection. In frequency dependent selection is the fitness of any one moth declines if it becomes too common in the population. Illustrates, notice this figure. Notice this is a research method using a virtual population to study the effects of selection. Application using virtual organisms that can reproduce and pass on characteristics to their offspring. Biologists can model the effects of selection over many generations in a compressed period of time. This method also enables researchers to isolate the effect of the study variable by controlling for other influences on the population's evolution, a nearly impossible goal in nature. So illustrates an experiment that simulates frequency-dependent selection in the wide blue jays look at and eat moths that sit motionless on tree trunks during the day. Captive jays can be trained to find virtual prey on a computer screen. These jays were presented with a series of screens with showed a speckled background, sometimes including an image of a moth. When a GA backed at a moth image, it was rewarded with a small piece of food. The GA soon learned a search image, a quick way to recognize the most common type of moth. As their skill grew, the computer program adjusted the frequencies of the virtual prey, making the common type rare and formerly rare types common, just as nature selection would do. The researchers also programmed mutations that introduced new moth patterns, whenever a type of moth became common, the GIS rapidly learned a new search image and targeted it. Frequency-dependent selection preserved polymorphism. In this population, rare moths of any pattern were at an advantage, and common moths of any pattern were at a disadvantage because the GIS learned to find the common moths more easily than the rare ones. Similar frequency-dependent selection has been observed in a number of predator-prey interactions in the wild. Back to modes of selection. Notice that first one. Notice also these cases describe three ways in which a hypothetical deer mouse population with heritable variation in fur coloration from light to dark might evolve. The graphs show how the frequencies of individuals with different fur colors change over time. The large white arrows symbolize natural selection working against certain phenotypes. Notice that this frequency of individuals represent, and this one, original population. Notice directional selection shifts the overall makeup of the population by favoring variants of the one extreme of the distribution. In this case, darker mice are favored because they live among dark rocks, and a darker fur color conceals them from predators. Notice also disruptive selection favors variants as both ends of the distribution. These mice have colonized Apache habitats made up of light and dark rocks, with the result that mice of an intermediate color are at a disadvantage. Notice that. Also, stabilizing selection removes extreme variants from the population and preserve intermediate type. types if the environment consists of rocks and of an intermediate color, both light and dark will be selected against. Okay, thanks.